lines, it acts as a roadblock to the enhancer. So the enhancer can't activate transcription of the gene. I have a little arrow that comes and blocks over here. Anyway, so this is known as imprinting, and the reason for it is not understood. What we know is those 200 genes that are being imprinted are usually genes involved in the growth of the fetus. And so there is a theory, and it's entirely only a theory, called the War of the Sexes. Okay? So many maternally imprinted genes, i.e. genes that are turned off by the mother, are genes involved in fetal growth. So the father has this thing. He wants to make sure his progeny grow rapidly and become healthy. That's the whole goal of males, right? Produce progeny and then walk away. So that's what's behind the whole story of the War of the Sexes. Okay? So the male's drive in evolution is to produce progeny. Female, on the other hand, is trying to conserve its strength for additional children that may be in line or by possibility. And so it's basically suppressing or repressing his growth genes. So it's preventing overgrowth of the fetus. So it's kind of distributing its resources. If you have a female producing 10 or 20 different progeny, it wants to make sure it provides progeny number 20 just as many nutrients as it does to progeny number 2. And so it kind of conserves its strength by turning off these genes that are involved in the growth of the fetus. Because as the fetus grows, it's going to absorb a lot of nutrients from the mother and deplete the mother's strength. And so this is a theory that the reason these genes are imprinted is basically a way to prevent massive growth of the fetus. Now, a lot of the syndromes associated with imprinted genes are syndromes that result in, because of a mutant version, result in overgrowth. Hear stories about fetuses that are overgrown and are very large. One of the reasons could be because of this defect in the. All right, so any questions? This is the last data slide I'm going to show you. Before I give you a 2-minute talk about something else. Enhancer from docking. So the way we enhance, here's a thing. Enhancers were discovered in the 1980s. I was a grad student when enhancers were discovered. And all we know about enhancers is they activate genes. But we don't know exactly how they do it. The enhancers are thousands of base pairs upstream of the promoter of a gene. We don't know how this enhancer finds the promoter of this gene. We still don't know. There are multiple theories. One of them is it actually walks along the DNA. Another is it hops along the DNA. Another is it just kind of randomly searches all over the place and when it finds it, it sticks to it. We don't know. So, irrespective, when you bind this protein, this enhancer cannot find it. That's all we know. Big gap in our knowledge about transcription. Very basic gap in our transcription, our knowledge about transcription. We fully know this after 30 years of this. So, I make things sound simple, but there are big gaps. But yes, that's exactly. We don't know how this works. It's just, we know it's a phenotype, it's a functional, but we don't know why and how it happens. Any other questions? Okay, first, thank you. Okay, so I'm going to talk to you a little bit. This is the end of the class. So it's been 10 whole weeks. And I hope, I really hope, I've given you a kind of panorama of genetics. Right? A big view of the world from my perspective. And I hope I've given you a foundation, a firm foundation, hopefully, for your future studies in biology. Because, as you all know, understanding genetics basically underpins a lot of biology. It underpins evolution, it underpins traits, it underpins enzyme function, you name it. Genetics underlies all of these different fields of study. You can study and use genetics to modify organisms. You can use genetics to alter the world. I'm a used car salesman for genetics. That's what I have to say, right? But there is a great book. There's a great book by probably one of the most provocative but forward-thinking scientists. He's in MIT. Name of George Church. Book is called Regenesis. You've got a summer break coming up. If you really want to think about the future of science and biology and genetics and its applications, read this book. But be prepared. There are scenarios in this book that are so phenomenally science fiction that they're scary to some extent. But I suggest you read it because it's a fun book to read. Now with your knowledge of genetics, you should be able to understand what he's trying to say and where he thinks the world is going to go with genetics. It's a story of synthetic biology, the future of synthetic biology that he describes in this book. And it's a really interesting book. What is it? George Church. So that's one thing that you have to keep in mind. You are going to read about new discoveries of genetics pretty much every week in the newspapers, leave alone in journals. And I hope this class allows you to evaluate those stories with some amount of knowledge about genetics, and you are able to kind of decide for yourself whether you believe the results or don't believe the results, whether you think they're good for the world or bad for the world. But I hope you do that based on knowledge rather than fear and fiction. And I hope this class kind of has taught you to evaluate this data in this way. Keep in mind that there is no such thing as a black and white world. The second book that I want to sell to you is a book by Keenan Malik. It's a history of philosophy and ethics. It has nothing to do with biology. And it's called The Quest for a Moral Compass. I read this book about a year ago, and I found it fascinating for the reason that what were considered ethical behavior by Socrates and the Greeks are not ethical today, and what we think of as ethical behavior today were considered unethical in those periods. And it's a history of not just the Western philosophical mind, but the entire philosophical mind, from the East to the West. So it's an interesting book. And it kind of, I'm selling this book to you because I want you to have an open mind when you read about science. I don't want you to kind of decide things based on faith. Decide things based on knowledge and fact. And make choices based on knowledge and fact, basically. So that's why I'm suggesting this one. Now, the reason I'm saying this is because genetics is a very powerful tool. You can do a lot of good with it, as we talked about in the class. We can identify disease traits. We can develop cures for diseases that have been uncurable until now. But we can also use the same methods to pretty much kill off the whole world. Yay. So it has the ability to do good, it has the ability to do evil. Does that mean that we stop doing genetics? Does that mean that we stop modifying organisms genetically? And that was a question we discussed in class, if you remember, about modifying genetics. And somebody came up to me later and said, you're basically proposing that we don't ban methods, but we regulate individual instances where they are being used. And that is exactly correct. it's much more easy to ban a method because you can just imagine yourself stopping everything in the path. But honestly speaking, that doesn't work. And once we go down that path of banning something, we're on a slippery slope. And so there's a third book, if you have time, called The Closing of the Western Mind. This is a history of the Dark Ages and how they originate. And when you read this book, you realize there wasn't one day, it wasn't like the Pope woke up one day and said, okay, we're going to stop all knowledge-based inquiry and we're going to believe everything that's based on faith. It was a very gradual process of small changes, small changes, small changes. And ultimately, around 1000 AD, led to complete elimination of knowledge-based inquiry in the world. And took us the Renaissance to get over it. And so I suggest we don't go down the path of just banning methods because we fear what those methods might do for us. And that brings me to my last slide. A proposition that was passed by Mendocino in 2004. And this proposition basically banned the use and development of genetic methods to modify organisms. So it was banning GMO. It was one of the first propositions to ban GMOs. And you guys, I'm hoping, know by now that genetics and genes are a function of DNA, correct? Everything is based on DNA. So, what is DNA? Deoxyribonucleic acid, correct? This is the proposition that bans the use of GMOs in any experiments in Mendocino County. And the authors of this proposition, by my view, have no idea about genetics or genes. Why do I say such a thing? Well, it's in the proposition. Here's the key sentence. Well, that's a section. It shall be unlawful for any person, firm, or corporation to propagate, cultivate, raise, or grow genetically modified organisms. Now, you know that we've been modifying organisms for thousands of years, genetically, and selecting specific traits. So, what do they mean? Well, they mean the use of modern recombinant DNA technology. Fair enough, recombinant DNA technology. So, what's their view of DNA? That's labeled right here. And this is their definition of DNA in the proposition, in the official proposition. DNA, deoxyribonucleic acid, means a complex protein.
No.
Now, at the stretch, you can say DNA is a complex carbohydrate because it's got sugars. But there is no amino acids in proteins. There are no peptide bonds in proteins. So, if you have people who are making decisions on your behalf but have no understanding of genes and genetics, do you actually follow them? That's the question I'll leave for you. Thank you. No definition. Okay. So for the exam, it's going to be 10 pages long. So 9 pages of exam and 1 page of the cover. 3 sections, multiple choice, short answers, long answers. There are lots of pictures, which is why it's an extra page longer than the midterm. Otherwise, it's almost identical in terms of the length. But you will have 3 hours to answer this instead of 1.5 hours to answer. Okay? So that's about the only difference. Now, keep in mind, everything we did from the midterm to now is in the exam. That includes little bits of epigenetics. I try not to put too much of epigenetics because you don't have as much time to study it. But everything else, fair game for me, including epistasis, including mutations, including pedigrees and RFLPs and restriction maps and cloning and whatever else. Okay? So everything else is good. Now, if you guys get confused in the exam with a question, please come and ask. It's not bad form to just ask for a clarification. It is bad form to ask for the answer for that question, but not a clarification on that question. So don't make a guess if you don't fully understand something. You could be wrong and you could benefit from asking. But anyway, all the best.